Okay, so I believe we'll go ahead and get started right now. Thanks to everyone who's joining us here today. This is the third installment of Andrews University Department of Chemistry and Biochemistry, our 2021 Spring Guest Lecture Series. My name is Desmond Murray, and I'm responsible for the series and this class out of the department. And a couple of business items here. We would like everyone to write their name in the chat with your location and affiliation possible. So put your name, location, affiliation. And very importantly, in the chat, that's all we want. For your questions, we do have a icon at the bottom that's called Q&A. Q&A is where you place your questions. Questions are not supposed to be in the chat. Questions should be in the Q&A. Okay. At this point, I will turn it over to Mike Nixon. And just one thing before Mike begins. Our seminar today is about 30 minutes of presentation and about 30 minutes of questions and answers. So that's the meat of our presentation and our seminar today. Again, thank you for joining. Mike.
Thank you very much, Dr. Murray. I'd like to welcome you all to this Zoom webinar. On behalf of our president, Dr. Luxon, and our provost, Dr. Arthur, I'd like to welcome you to this Andrews University Zoom webinar event. And thank you for being here. I'd also like to give a special thanks to Dr. Murray, who you just heard from, who was an integral part in putting this program together. And we are excited to hear from Dr. Corbett today. Before I pray to open us up, I just wanted to share a few quick words to set the table for our discussion today. This past Tuesday on January 26, 2021 was an interesting milestone for our campus. One of our university communications personnel, Stephen Payne, pointed out to us that it was on that date, January 26, 2020, that we sent our first communication to the Andrews University community regarding COVID-19 and the fact that we were monitoring the COVID-19 pandemic. At that time, there were 5 confirmed cases of COVID-19 in the United States. But on that date, this past Tuesday in 2021, we sadly had over 25.3 million cases on that day in this nation, and of course, over 421,000 deaths. What a difference a year makes, indeed. This past April, we also talked about the chilling effects that COVID-19 was having, in particular on communities of color, and we continue to wrestle with and combat this pandemic that we are all going through. And so we are excited to hear from Dr. Corbett about how the COVID-19 vaccine and the different vaccines that are available are another important tool in helping us to combat this pandemic. And so we are really excited to hear from her, and we are hoping that in the next year to come, those numbers will start to change for the better. Pray with me. Dear God, we thank you so much for this day, for this opportunity and this time together to hear about this really important tool that you have used, Dr. Corbett and other experts, to help develop these COVID-19 vaccines to help us deal with this very aggressive and in some cases deadly disease. We pray, God, that you be with everyone who's been affected by it over this past year as we reflect on the journey we've been on as a community, but as the same journey that our nation and world has been on as well. We pray that in the time to come that these vaccines will be able to get out to as many persons as possible in a safe and effective manner to help us to curb the effects that we've seen over this past year. And we pray that the best is yet to come in regards to mitigating the effects of this disease. In your name we pray, amen. At this time, I'll turn it over to Zoe to introduce our speaker.
Good evening to all. My name is Zoe Gentles, and I am a sophomore chemistry major at Andrews University in Bering Springs, Michigan. Today, I have the incredible pleasure of introducing our seminar speaker, Dr. Kizzmekia Corbett, a trailblazing woman of science and faith. Dr. Corbett is currently a viral immunologist and senior research fellow at the Vaccine Research Center at the National Institute of Allergy and Infectious Diseases. Dr. Kizzy Corbett was born in Hurdle Mills, North Carolina. In high school, she was part of Project SEED, a summer research internship program run by the American Chemical Society that exposes high school students, particularly from disadvantaged and underrepresented communities, to careers in chemistry. This early exposure to authentic research was the beginning of her remarkable journey in science. Dr. Corbett graduated from the University of Maryland, Baltimore County with a Bachelor of Science in Biological Science and Sociology. As an undergrad, she also researched at the National Institute of Health. She continued her studies at the University of North Carolina at Chapel Hill and received her PhD in microbiology and immunology. During her doctoral studies, Dr. Corbett researched human antibody responses to the dengue virus in children in Sri Lanka. The same year Kizzy received her PhD, she became a research fellow at the National Institute of Allergy and Infectious Diseases Viral Pathogenesis Laboratory, where she works to this day. Before the pandemic, her team's research focused on the understanding of fundamentals of the coronavirus, specifically research dealing with severe acute respiratory syndrome, also known as SARS, which is very similar to COVID-19. Soon after the first case of COVID-19 was confirmed in December of 2019, her team redirected the research to create an mRNA vaccine to prevent infection by the COVID-19 virus. Dr. Corbett says that her team engaged in this research in a fast, reliable, and universal way. To conduct human trials and eventually manufacture the vaccine, her team partnered with biotechnology company Moderna. After successful human trials, the FDA approved their vaccine on December 18th of last year. 4 days later, the first doses of the Moderna vaccine were distributed and administered in the United States. Now that the vaccine is available, Dr. Corbett has been directing some of her time and focus to improve the public's trust in science, research, and efficacy of the new COVID-19 vaccine, especially in underserved communities. This is part of the reason she accepted our invitation. Healthcare disparity has always been an area of concern in the United States and has become more evident due to this pandemic. There needs to be a significant push to develop national strategies to help address inequality in healthcare and as well as hesitancy to receive the COVID-19 vaccine. So I am excited and honored to welcome you, Dr. Kizzmekia Corbett, to our lecture series, our university, on behalf of everyone who was here to listen to you. Thank you.
Thank you so much, Zoe. And I almost feel like I don't even need to give my lecture because you did your research. Some of my taglines were embedded in that introduction. But yeah, you know, I'm happy to be presenting to you guys today. And I actually have been foregoing, at least for the current time, any presentations to academia because I feel like it is my duty now to talk more to the community, especially as the vaccines are rolling out and people have questions. And so it goes without saying that a university that is faith-focused took heed to that in a way that I am very much appreciative of. And so this presentation serves to give a backdrop of the science, but then to tell really the story of the rapid development of the safe and effective COVID-19 mRNA vaccines really with a kind of community-facing agenda so that I can help anyone in the audience to understand how this vaccine development came about over the last year. And while it looked rapid, how much work went into it prior to the onset of the pandemic. So without further ado, I am Kizzmekia Corbett, and I am the scientific lead of the coronavirus vaccine team at the Viral Pathogenesis Laboratory at the Vaccine Research Center, which is this building in the background of this slide, situated at the National Institutes of Health. So coronaviruses actually always pose as a pandemic threat. This family tree on the left-hand side of your screen shows two subfamilies of coronaviruses. The first of which are alpha coronaviruses, and the second, beta coronaviruses. There are 7 coronaviruses that are known to infect humans, 4 of which that sit in the alpha and beta coronavirus subfamilies that cause endemic or so-called seasonal coronavirus disease. So that's like the sniffles during flu season. Then there are the other coronaviruses that are zoonotic pathogens that have been transmitted to humans and caused epidemics. The first of which was SARS, which has to date tallied over 8,000 cases worldwide. And then MERS, which has to date tallied over 2,000 cases worldwide. It goes without saying that history continued to repeat itself. But one thing that we also understood from a laboratory perspective by way of a lab at the University of North Carolina is that viral evolution showed us that there were SARS-like viruses that were poised for human emergence. And so I like to say that the 2019 emergence of SARS-CoV-2 that turned into a global pandemic, now with over 100 million cases worldwide, is really the virus's way of saying, I told you so. We are currently in our 3rd wave of COVID-19 in the United States. This graph just shows the 3 different waves, the first of which started in the early spring of last year, and then another that started in late summer, and then this last one, which you can see the slope of this line starting in late fall around the time of the holidays, continues to rise. And so I show that for the United States, but then I also show it for locally here in Maryland where I live. So it's estimated that about 60 or 70% of population immunity must be established in order for us to control COVID-19 and start to get back to the point where we can resume some of our normal activities. With that, if immunity was solely dependent on a natural infection, then about 40 or 50 million people would have to succumb to COVID-19 disease worldwide, which is absolutely unacceptable. So at this point, a vaccine is needed to prevent the dynamics of the pandemic.
A global race to find treatments for the sick and a vaccine to protect the healthy.
This ends with most or all of us being immune to this virus, and ideally that's through a vaccine.
Can solutions that usually take years,
we are trying to move as fast as we possibly can,
be found in a matter of months?
It is an extremely revolutionary approach to get into a phase 1 clinical trial 66 days after the sequence of the virus is released is a world record.
And so how did we accomplish this so-called world record? The way that we did this was by prototype pathogen approach to pandemic preparedness. So we knew that coronaviruses posed as a pandemic threat to the globe. And so the idea behind this approach is that you would study coronaviruses by way of a prototype virus within that family. We chose the MERS coronavirus, which is closely related to SARS-CoV-2. You would generate enough knowledge around that prototype virus so that you can apply it to the viral family in all. The second prong of this approach would say to develop that countermeasure, namely here vaccines, through phase 1 clinical trial. And this allows for us to speed up the development of that countermeasure for other viruses within that family. And while we didn't necessarily get there for MERS coronavirus via way of this approach, what we did do is, as I called it in the New York Times, had a way by which we could plug and play our approach for MERS coronavirus vaccine development into what we were doing for SARS-CoV-2. The outline of this approach is really well detailed in the Journal of Clinical Investigation by my principal investigator and I. And so if you're interested, you can take a look. So vaccine preparedness for the pandemic threat of coronaviruses. So knowing that we were in threat of emerging coronaviruses, we knew that we needed fast, reliable, and universal vaccine solutions. What does that mean? For fast, vaccine technology that must be able to be produced in vast quantities quickly. Reliable, vaccine technologies that must be produced and uphold a reliable manufacturability standard. And fast. Universal. And I have this italicized because universal vaccines are thought of as those that are preemptive or that provide protection against future outbreaks. And while we're not completely there yet for coronaviruses, in the italicized version of that, we're at least ready and preemptively, with a preemptive way of making a really fast and reliable vaccine. So what would a COVID-19 vaccine do? There are two sides of the story. The first of which we all are very well aware of, is the benefit to us and on our individual basis. So that is, if I take a vaccine, there is reduced severity of illness and perhaps prevention of infection. The other side of it is the benefit to the community, where a vaccine can reduce transmission and then ultimately start to make it safer for us to resume our normal activities. And so as I tweeted, and if anyone follows me on Twitter, you're probably aware that I tweet a lot of my thoughts. One of my thoughts is that vaccination is community service. And let me tell you why. It is because of this concept of population or so-called herd immunity, where the stick figures on this slide in the non-immune population are blue, and in a population that is partially immune, the people that are immune are green. If you were to release a virus in the non-immune population, you can see that there is high transmission. So many of those stick figure people turned red and got COVID-19. If you are to release that virus in a partially immune population, where the blue people were the only ones that were able to get infected, you can see that there are only a scattered amount of people who got COVID-19 or that turned red, and that means that there's low transmission. The other interesting thing that's a side note from this is that you can see that those people that are red are spaced further apart. So you can see as you add green people in the population, the virus continues. The virus in so many ways has roadblocks and the transmission starts to be impeded. Now, the rapid timeline for SARS-CoV-2 mRNA vaccine development. We saw that the sequences were online on January 10th. The virus was then called 2019 N or novel coronavirus. The Chinese government graciously posted those sequences to the public. In 66 days, as was said in that video, we in a world record speed got into a phase 1 clinical trial. Now I know what it looks like. It looks like Operation Warp Speed, and it looks like there could be levels of irresponsibility in this development process. However, what a lot of people don't know is all of the work that went into the vaccine development for coronaviruses in general through the years from 2013 all the way into the onset of this pandemic. And so let's talk about that work. The coronavirus spike protein is the protein that sits on the surface of viruses and is the reason why the viruses get inside the cells. So the coronavirus spike protein grabs onto a human cell receptor. In the case of SARS-CoV-2, that is the ACE2 receptor. By that interaction, it allows for the virus to get inside of a cell. And so if you want to have an effective vaccine, you want to, at the very least, impede that reaction of the binding of the spike protein to the human cell receptor so that you can stop or block infection. And so when I came into the laboratory at the Vaccine Research Center, we knew nothing about how those coronavirus spike structures are looked in any real high definition. And so with that, we started to study this coronavirus spike proteins in very much detail, where we were able to publish the first structure of any human coronavirus spike protein. And that paper was in Nature and really showed us on a very atomic level what those proteins look like so that we can design really good vaccines to target them. At the same time, another coronavirus spike structure was published, and then there was really an onslaught of literature detailing different coronavirus spike proteins, their functions, and etc. What was missing from that literature, though, is while we were able to define the structures of those spike proteins across several laboratories, we were the only ones that took it a step further and began to start to understand the immunogenicity of those spike proteins. So that is, how well do those spike proteins cause immune responses? And we did this by way of rationally designing a MERS coronavirus spike antigen, or the thing that is given in a vaccine. We did this via a method called structure-guided stabilization. And so these proteins are fusion proteins. And so what that means is that they allow for the viral membrane and the cell membrane to fuse together in this very intricate set of steps. Long story short, there are two really distinct versions of these proteins. The first of which is pre-fusion, so before fusion occurs. The second of which is post-fusion, after fusion occurs. The most functionally relevant to the body is obviously the pre-fusion part of the pre-fusion form of the protein. And so you want to target antibodies specifically to the pre-fusion form of the protein. And so in order to do that, because these proteins have a spring that sit in the middle of them that allows them to unleash and fuse and cause fusion, you have to lock that spring into its pre-fusion conformation. And so we scanned the area of these proteins around this FP, or the fusion peptide, and we begged the question, what types of mutations would allow us to lock that pre-fusion protein, that protein in its pre-fusion state? We stumbled across what is now called the 2P mutation, where expressing MERS spike with the 2P mutation gave you this beautiful blue line. So it's a beautiful homogeneous mixture that supersedes that of the MERS S wild type that you can't even see on this graph. So that means that it expressed better. And when you looked at that protein under the microscope, it looked homogeneous and in its pre-fusion or mushroom-like structure. Whereas in a wild type form, it flipped readily into its post-fusion conformation. And so we asked the question, well, if those two protein mutations work for MERS, do they work for other coronavirus spike? And the answer is absolutely yes. Published in that paper were the HKU1 S2P alongside the SARS S2P and really featuring the MERS S2P. We also dropped those mutations to effectively stabilize multiple coronavirus pre-fusion spikes. Interestingly enough, also the SARS-like spike that the viral evolutionary biologists termed to be potentially emerging. So now we had the protein. We did animal experiments, and so we knew that it elicited really, really good antibody responses, and even though I don't show that data. But if you're going to be developing for a pandemic, one of the things that you need to do is be fast. And so making protein in the laboratory is not always that fast, especially when you're making clinical grade protein. And so what we wanted to do was turn the body into the protein factory. And we did this by collaboration with Moderna, where they use messenger RNA to deliver the message of any new therapeutic protein. Here, we're delivering the coronavirus spike protein. They take that message and they encapsulate that message and formulate it into a lipid nanoparticle, so a very small ball of lipid. Given that very small ball of lipid with the message inside, you inject it into the muscle, and then the muscle cells read that messenger RNA, and then it starts to make spike protein, because that's what cells do. They read messages and they make the proteins that they're told to make. That spike protein is foreign to the body, and so the body starts to learn in very intricate detail what it looks like, in so much detail that it remembers what it looks like, and that immune response can hypothetically be protective later. The cool thing about the mRNA platform is that it's fast and it's reliable, because it upholds a manufacturability standard that has been tried and true, and it's universal enough in that you can really plug and play whatever protein you want into the backbone of it. And so we tested that concept using the MERS spike with the 2-proline mutation, delivering it by messenger RNA in collaboration with Moderna. Basically, what we showed is that you can deliver that MERS S2P mRNA to mice that can succumb with MERS challenge. So if you give these particular mice a dose of MERS virus into their nose, they die. Whereas when they're vaccinated, they do not, or they don't even really lose any weight, except at this very, 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 very low 0.01 microgram dose, which is almost nothing. It wasn't just that the weight loss curves showed promise, but also that the lung viral load and the amount of disease by way of lung hemorrhage showed promise as well. So we were protecting mice from dying and from having lung disease in a dose-dependent fashion by giving the MERS spike protein via messenger RNA. So we knew exactly what to do because we tested it even down to the doses that were needed in small animal models using MERS as our prototype pathogen. So we had the protein, we had the messenger RNA, and we had established a collaboration. And we had a really cool understanding and confidence that we would elicit really good immune responses if we did it the right way. So therefore, when the sequences came out on January the 10th, 4 days later, Moderna was able to initiate clinical grade production of the mRNA 1273, which is the vaccine sequence that we came up with based on our previous knowledge, obviously encompassing the 2-proline mutations. Moderna was able to ship that clinical drug product to the phase 1 clinical site in only 41 days, which really is a testament to the utility of messenger RNA as a very rapid manufacturable and scalable platform. The phase 1 clinical trial started on March 16th, 2020. And although I don't detail it in this brief presentation, there were several steps in between there that a lot of people don't even know about because that's not what the media talks about. These steps where we were testing the product in preclinical phases in mice and monkeys are the reasons why we were able to move so quickly into an FDA safe to proceed a vaccine trial. All of these steps are detailed in an article that I wrote or that we wrote in Nature, talking about how we used our prototype pathogen preparedness to design the vaccine and test it in very, very lots of detail in mice. And so let's talk about the phase 1 clinical trial a bit. We actually did two, or we started with two, one of which was in Seattle, where we tested mRNA 1273 in young adults 18 to 55 at 3 different doses. The second of which added on elderly people at 2 doses of 25 and 100. The data that you're mostly going to see today are from the 2 doses of the vaccine, just like you get when you get your vaccine, following those people through to 2 weeks following their second dose. What we showed is that mRNA 1273 elicits really potent neutralizing antibody responses in young adults. Those young adults are shown here in the green, where 18 to 55-year-olds got 100 micrograms, which is the dose that you will get if you do so choose to get mRNA 1273, and they elicited really robust 10 to the 3 neutralizing antibody titers. Looking at the people that were older, between 55 and over 71 years of age, you can see that the same remains true. This is after the first dose, where there's a blip in neutralizing antibody responses, but really after the second dose, so you see this whopping neutralizing antibody response that is on par with what you see in the young adults. And so that was actually very, very promising. As with previous vaccines, like for example, the seasonal flu vaccines, vaccine-induced immune responses in older people are a little bit dampened. We also have, to this point, published data out to day 119 in those phase 1 participants, showing that the antibody responses up to day 119 are not waning significantly, which shows that these antibody responses are not just robust, but also persistent. We went into a phase 2 clinical trial And while I won't show any of that data today, that phase 2 clinical trial allowed us to test 2 doses, 50 micrograms and 100 micrograms in about 600 people, and it was sponsored by Moderna. Obviously, as everyone knows, mRNA 1273 is now approved for emergency use authorization by the FDA, and that is by way of this randomized placebo-controlled phase 3 vaccine efficacy trial. So to explain the trial that was conducted by Moderna is that there were 30,000 total people, 2 groups. One group of people got the vaccine, another group of people got the placebo. After they got dosed with mRNA 1273 or salt water, they went off into their normal lives, and they were able to, if they got any symptoms that looked like they might be COVID, whether it be a headache or a sneeze or otherwise, they contacted their trial physicians and got diagnosed as a COVID case or not. As time accrued over the course of the vaccine study, the cases were tallied. And how you get vaccine efficacy is by asking the question, how much percentage more of COVID cases were in the placebo group as opposed to the vaccine group? Taking these stick figures as an example, in the vaccine group, if 2 volunteers got COVID-19, whereas in the placebo group, 18 volunteers got COVID-19, you have a 90% efficacy because you have 18 of your 20 total people that got a COVID-19. So the vaccine, getting a vaccine decreases your chance of getting COVID-19 by 90%. The phase 3 clinical trial actually had some really cool demographics, and I do want to just talk about these only for a little bit because it's important for people to understand, especially in the general public, that these vaccines were not just tested in your standard white male, but they were tested amongst people who have really represent the demographics of the United States. That being said, as far as race is concerned and ethnicity, about 10% of the phase 3 clinical trial participants were Black or African American. Another 4 were Asian American, and then 20% identified as Hispanic. With comorbidities such as diabetes, sitting here with a really large 36% of people having diabetes in the clinical trial, obesity, and etc. So there was a very intentional effort to enroll people who were both racially and ethically diverse, and also who had a diverse subset of COVID-19 comorbidities so that we can study the vaccine in those subsets of people. Looking at the efficacy and safety of mRNA 1273 in the phase 3 interim analysis. So what does interim analysis mean? That means that people got 2 doses of mRNA 1273 or placebo. The placebo is the purple line, mRNA 1273 is the orange line. There's 1 dose with an arrow, and then there's a second dose with another arrow. After those doses, about 2 weeks after the second dose, the cases started to be counted. And as you can see, as time passed, people in the placebo group got more and more cases, whereas people in the mRNA 1273 group did not. All in all, as at this interim analysis, there was about 11 people in the mRNA 1273 group that got COVID-19, whereas 185 got symptomatic COVID-19 in the placebo group. That's where you get altogether your 94.1% efficacy. Interestingly enough, you can see here in the severe COVID-19, none of the participants in the vaccine that got the vaccine had severe COVID-19 as characterized by hospitalization. Whereas in the placebo group, about 30% of the, about 30 total severe COVID-19 cases happened. Thinking about the side effects, as we call it, but as the FDA calls it, adverse event, after the first dose, the most significant of the adverse events are pain or swelling or redness at the injection site. So you can see that about 84% of people get 1 or more of those at the injection site following their first dose. There's a little kick after the second dose, and I can even admit that myself because I had my second dose on Monday, and I had fatigue, which is one of the symptoms that people generally get, and also a slight headache for about 6 hours, and also still too some pain at my injection site. Nevertheless, about 80% of people who who got mRNA 1273 compared to 35% of people who got the placebo had one or more of fever or joint pain or muscle ache, which actually just tells us that the vaccine is working in the way that it should, because it's really igniting your immune system to act very rapidly. Point that I must make, because I am talking about a messenger RNA vaccine, is that there is no way that this vaccine will alter your DNA. There is no way that this vaccine will alter any of your fertility. And as well, in this phase 3 clinical trial, there were no deaths that are attributed to mRNA 1273. And further, to date, there have not been any deaths that have been specifically attributed to vaccination outside of comorbidities in the general population. Giving a brief overview, just quickly, of the total global vaccine pipeline. So there are about 200 vaccines in the global pipeline altogether, most of which are still in their preclinical stages, which again goes to show that that's where we really soaked up a lot of time over the last 6 years and allowed us to move very rapidly into the phase 1, where there are currently 36 vaccines in phase 1 clinical trial to test their safety and doses. Phase 2, 14, and then phase 3, 11. Currently around the globe in limited use are 6 vaccines, 2 of which are in limited use here in the United States, and none that have been approved for full use. Honing in on the phase 3, the vaccines that are in phase 3 in the United States only, I talk a lot about the Moderna vaccine candidate. There's also the Pfizer-BioNTech vaccine candidate, which is very similar to Moderna's, aside from the fact that it's a 30 microgram dose and it gives, you get your doses 3 weeks apart as opposed to 4 weeks apart, but very similar vaccine candidate. Then there is the Johnson & Johnson vaccine that uses the spike protein, but it delivers that by an ad vector, which is an adenovirus vector. So it takes out all of the replication machinery of the actual adenovirus, puts in the spike protein, so it exposes your body to a just normal cold virus, the advirus, but also gives you immunity specifically towards the spike protein. AstraZeneca uses a similar technology with their ChAd ad vector, and then Novavax is giving the spike protein in its prefusion conformation by way of a nanoparticle with an adjuvant called matrix M. Altogether, one of the things that stands out on this slide are the words prefusion stabilized. Remember that we defined that prefusion stabilizing mutation that is being used in 4 out of the 5 vaccine candidates that are being streamlined in the US for development. Altogether, though I didn't show many of those data, these vaccines have shown efficacy in small animal models and non-human primates. They elicit robust neutralizing antibody responses and even some T-cell responses. The front runners have promising interim efficacy results, as we've seen with the 94-95% efficacy with Pfizer and Moderna, and then also with about 60 to 90% efficacy in interim analysis for the AstraZeneca candidate. With that, I like to take the time to thank my laboratory, particularly these people in the middle, who have been working with me for the last six and a half years, really fundamentally trying to understand vaccine development for coronaviruses, and especially my mentor, Dr. Graham, who, as you can see, slid to the side in this photo and all-encompassing to allow me to lead this team in a very independent way, and almost selflessly of him. The COVID-19 response at the VRC has now become a VRC-wide effort. All of the people on this slide have absolutely helped tremendously, whether it be communications, human resources, animal experiments, etc. Since the beginning of all of this, we've been collaborating very strongly with Moderna, who manufactures these very brilliant messenger RNA vaccine platforms. But also for structure, we collaborate with Jason McClellan's laboratory and the Scripps Research Institute with Andrew Ward's laboratory. Our mouse challenge studies have been conducted at the University of North Carolina at Chapel Hill, when Ralph Barrick's laboratory, largely led by Sarah Lice, in the early days by Adam Cockrell. Then we also have a veterinary pathologist, Dr. Ian Moore at the NIAID, who has been of tremendous help in understanding lung pathology in these vaccinated animals, or lack thereof, I guess. And then a special shout out to all of the people who have just given their trust and their muscle to the development of these mRNA vaccine candidates, not just Moderna's, but Pfizer's, and for the people who are currently enrolling into the Novavax trial as well. And with that, I will take questions.
Okay, Dr. Corbett, we have a question from Howard Bailey. Howard wants to know if there's any way of knowing how effective the current vaccines and vaccine candidates will be against the UK variant of the SARS-CoV-2, which has 17 mutations. Any thoughts about that?
Yes, that I posted actually on Twitter a thread about it, and we think that we are perfectly okay. We published data looking at the pseudovirus neutralization of the vaccine sera to these mutants or these variants, so both the UK and also the South African. Actually, the South African one is actually more alarming when you think about spike structure. But nevertheless, we still elicit antibody responses towards the variant. I say that with the disclaimer that obviously one of the things that we want to do on a population basis is to stop this virus from transmitting so that we can stop the induction of new mutations into the spike protein. But nevertheless, for now, it seems like we will be good.
Okay, so I think that touches on one of the more popular questions as well, that a lot of people have been asking: is this vaccine potent enough to deal with the mutation of the virus?
Right, that's what I think.
You did, but just wanted to say that, yeah.
Okay.
That's a question that people are asking a lot, not only here online, but also in real life. Okay, so another question is, are there other scientists who have different perspectives on the safety of the vaccine?
I'm sure there are other scientists who have different perspectives on every single drop of the vaccine, because that's what scientists do, is they criticize each other. But all the scientists that I know are very much in understanding that once we saw the phase 3 data, that the phase 3 data showed that the vaccine was safe and effective. And because at least reputable scientists understand how messenger RNA works and how quickly the message disappears, just like a Snapchat message on your phone. It doesn't stay in your cell forever. It does not travel to your nucleus of your cells and can't do anything with your DNA. And also that messenger RNA has been in people for years, not just with this vaccine, but for years. I think that most scientists who are well-read understand that this vaccine is safe.
And as a follow-up to that question and your answer, I'm thinking also that obviously within the scientific community, there is the peer review. And so a number of scientists have gotten to review the work. And I believe, didn't the CDC or FDA put out the entire report for everyone?
Right. So the entire report, even for the phase 3, the entire report was published online for everyone. And also the debate about the data, I guess you can say. It wasn't really a debate because the data was very clear, but the advisory committee to the FDA around vaccine safety and efficacy met, and that meeting was public. I actually watched it. I took off that day in so many ways and watched that meeting. And so this has been a fairly transparent process. And also to the point of the reviewers, I mean, for example, the mouse paper, when you go to Nature and look at that paper, you can see the reviewers' comments. And so you can see how different scientists think about vaccines, but also how we were very diligent with making sure that we answer all of those questions with lots of forethought.
So just to wrap that up, when in the scientific process, this is part of the process. Okay, so the
It
idea
is
is
why
like,
it is why my eyes are like this, because I'm writing right now, and
yes. So
so
it's not,
so writing up your data, and having other scientists who are world-renowned scientists and take their time generously to peer review your data. They look for flaws at every single corner of the data, experimental design and statistical analyses in every single corner of the data. So that is just an embedded vetting process of any science, not just vaccine science.
exactly. And I think that's a good thing for people to know generally, that in the scientific process, that sort of back and forth and helping each other in terms of making sure the data and the interpretation of the data is correct.
Right.
Okay. Next question.
All right, I've got another question for you, Dr. Corbett. Someone wants to know about transmission of the virus after getting the vaccine. You've done a really good job illustrating how the vaccine actually protects us with herd immunity and slowing down the spread and so forth. But once you have the vaccine, what are your thoughts on, or do we have any data that speaks to whether or not we can transmit it?
I'm so glad that you changed the way that you phrased that question.
Yeah.
It talks to the fact that you're a scientist, but never mind. Because like people say, do you have any thoughts? Of course I have thoughts, but I don't spew my thoughts into 500 people's ears in the middle of a pandemic. And so what the science says is that we just don't know how well this virus will prevent transmission from human to human. That is because, right, how you're transmitting virus is by harboring replicating virus in your nasal pharynx. What was tested and by way of the phase 3 clinical trial for efficacy was around disease. And so that is in your lower airway, which by all accounts is a little bit easier to protect. And so until we have those data in full around transmission, whether they come from phase 4, so to speak, or this rollout to the general public, or from animal models, we just can't say. And so the advice to continue to wear your mask remains true. That being said, embedded in our New England Journal paper for the non-human primates, we show that vaccinated monkeys that got the clinically relevant dose, so that is the 100 microgram dose, rapidly clear in 2 days, actually like less than, probably 1 day, but we tested 2 days. In 2 days, they rapidly clear the virus from their nose, as opposed to the animals that did not get vaccinated, obviously did not clear the virus from their nose. So maybe based on some animal experiments, but we just don't know yet.
Okay. So there are a couple of questions that are interesting. Yeah, what are the ingredients of the first vaccine? What are the ingredients of the second vaccine? People are assuming that there might be a difference.
I mean, subtle differences, but nothing to be any concern. There's no formaldehyde, there's no fetal cells, all of the things that the internet claims there to be. There are not even any preservatives in these vaccines, and that's why they have to be stored at these very low temperatures. So the vaccine is messenger mRNA, so genetic code by way of a message, and lipid, fat, so a fat ball, some sugars, and some sugars, sucrose, so some sugars. It's the list of the materials. I should actually put that on a slide. That's a good point. But the list of materials are online, and they are as pure as you're going to get.
Okay. Next question.
All right.
That's actually the beauty of letting the body be the protein factory.
Right.
Because you can deliver fat. You can deliver a genetic code in fat and sugar, and the body just takes care of the rest.
So you just answered another question. Sorry, Barbara.
No, that's all right.
People were talking about how do DNA treatments differ from classic vaccines?
Well, this is not a DNA. This is not a DNA
treatment. Not DNA, sorry, RNA. How do RNA treatments differ from classic vaccines?
So This classical vaccine is depending on how you think of classic. So historically classic, meaning that a virus is inactivated or weakened and then given to the body, so that you can't really make any replicating virus, so to speak, and you don't have a robust infection, but it does alert your immune system and tells your immune system how to look out for that same virus again. So that's the classical way, classically historical way. There are other ways like protein. So taking, for example, that spike protein that I showed on my slide, making that in a very clean, safe laboratory and in a production facility, and then delivering that protein into the muscle is another way. Or you can do it this way, where you skip all of that and you just put the genetic code for the protein into the muscle and have the body cells make the protein instead, which is basically what happens in a lab, right? You put the protein genetic code into some cells in the lab, and then you let those cells spit out all the protein, and then you purify the protein. And so we just skip that step. And that's the difference.
All right, I've got a question here for you. I think I know the answer, Dr. Corbett, but I want you to share it, because I think this is a common question that many people ask. If we have so much information on viruses and vaccines and so forth, why isn't the common cold cured yet?
Well, you know, we get these questions a lot. It makes me sad a little bit, because it does. We get it around HIV, we get it around certain cancers, and it makes me sad because I think that I just feel sorry for the people who have invested their entire lives trying to make vaccines for these things. Like my boss, who is 35, 40 years into his career, and really just now getting to the point where he saw something through a phase 3 clinical trial. And the reason is because things are not monoliths. The common cold can happen by way of several different viruses, whether it be influenza, rhinovirus, adenovirus, and et cetera. And you have to learn, in order to cure so-called the common cold, you would have to basically vaccinate against all those things. And these viruses are different. They don't look the same to the body. You're not going to be able to trick the immune system in the same way for every single virus. And so it's a learning process. And that learning process is what we did for the last 6 years for coronaviruses. And so the viruses are not monoliths, and that's one thing. And then the other thing is that we are really getting to the point where technology is advancing so much that we're transforming vaccinology in a really cool way, whether it be from I'm like on like zero sleep, so sorry, but seriously, for like the 200th day of my 2 years, or last year. But yeah, so there's the messenger RNA side of the transforming vaccinology, but then also the thing that we did around understanding in high definition the spike protein, so we could learn how to make a good vaccine. That technology is something that's also being revolutionized. So technology is coming, and things I think will start to change. But I think to say, well, if you know so much, why don't you do it for everything? Because I'm sleepy. and so is every single scientist in the world, especially I'm just now to the point of 34 years old, but like many of these scientists, especially in respiratory pathogens, have worked for years, right? And they've done so much, and their work actually fed into this vaccine development process in more ways than one. And so we'll get there. But I think I'm going to be employed for a while because we have a long way to go.
Okay. So I know one of the things that's important to you is talking to people about trustworthiness, especially to persons who have a hesitancy towards the vaccine. And so one of the questions that's being asked is, what do you think is the best approach to talk to non-scientists about vaccine hesitancy? This person is struggling because I lose them when I get too scientific.
Yeah, that's the best approach is, so you're not going to really beat vaccine hesitancy with just facts, spewing facts at people, right? People aren't dumb. The facts are online. The publication, people can Google publications and like all these things. I know it's very hard to decipher, but at the end of the day, if people wanted to just find facts, they could go find them online. What people are needing, I think, not just around vaccine hesitancy, the world just needs empathy, period. You have to like place yourself as into a non-scientist's shoes for just a quick second, understanding that what people just saw is like something that's completely historic, absolutely revolutionary. In 10 months from sequence to an EUA approval. And nobody outside of, people who study coronaviruses and a few other select viruses knew what was going on for the last 6 years because they followed the publications. And so I think that one of the things that has been helpful for me is that that's what I tried to do today. I just try to put people in my shoes over the last 6 years, walked you through the process, told you, these are the questions that we asked and this is what we did. And these are the steps that you didn't hear about because they don't talk about mouse studies on CNN. And those kinds of things. I think it's important to just put people inside the process and make people feel like a scientist for a day, but not by saying, well, actually, you're wrong. By saying, I understand how you could think that, because it's rational that one can hear RNA and think that you could change your genetic code. I mean, like, outside of a scientist, it's so rational to think that. I understand why you think that, but actually, the way that cells work with DNA as opposed to the way that cells work with RNA is completely different. And so they don't even come in contact with each other in the same places of the cell. And just saying those kinds of very easy things to people. And the other thing too is that it's vaccine hesitancy is not a monolith, as the same, right? So you really almost have to target vaccine hesitancy on not on a one-to-one individual basis, but really on a community-to-community basis. So my uncle, who is a tobacco farmer in rural North Carolina, surprisingly enough, this is my uncle who like has known me since birth. My boss being from Kansas and being like from a farm in Kansas, and the fact that, the father of vaccinology, Maurice Hillman, was also born on a farm and raised on a farm. That helped my uncle to like come across to the vaccine more than me being his niece, right? because to be frank, I speak a very different language than a tobacco farmer in North Carolina. And so and then it's just that kind of thing. So you have to individualize. And the current concerns are often also very individualized because, for example, I'm seeing a lot of things in this chat about natural remedies. And so we have, fortunately, for all of our healths, including myself, I eat healthy as well. Not a vegan, but I am, pescatarian and take vitamins and drink some seaweed. Great. But natural remedies are not going to prevent you from being infected with the virus that causes COVID-19 or any virus on a specific enough level that you decrease your chances of being sick by 94.1%. And that's just that, right? If that were the case, then I promise. Number one, I would take a nap. And then number two, I would certainly be like rallying behind selling every single free giving away natural remedies. Because that's just the facts, right? Natural remedies are not building up specific antibody responses. What they are doing, especially in your normal healthy person, is just making your cells healthy enough so that you might be able to be, less affected but you still might get sick. You might just,
Right.
you know, only have a cough as opposed to being on a ventilator. And also what's important is that for supplements, when you see the data for supplements, not around COVID, because there isn't really much out, but for other viruses, supplements are supplements. If you are a person who starts here on the right level for whatever vitamin it is that your body needs, adding to that is not going to decrease your chances of getting really sick. It's only when you are deficient in something that supplementing your body helps you to prevent illness. And that's because your cells were starving for a vitamin or a supplement. And so now you've boosted your ability of your cells to just act. Whereas a normal healthy person, your cells are already just percolating, ready to go. And so I think that's really just a really this thing that we just have to really understand. And I rally behind exercise and being healthy and, you know, drinking your water and like all of these things. But vaccination is a completely different ball game. And we are not going to be able to seaweed and supplement our way out of a global pandemic.
So that to me brings up another point, piggybacking on some of the comments you just made, is that vaccines are not, however, the only strategy out. Could you speak to the fact that we still need to be researching and developing treatments?
Right.
Therapies?
So, you know, we don't even talk about this, but here's just
Right.
in that
Right.
our team actually developed the first monoclonal antibody therapy. So the Eli Lilly therapy, that's our intellectual property.
Okay.
So we think about therapies too. we just don't necessarily talk about them. And the reason is because therapies work for populations of people who they work in the interim while vaccines are being developed because they're generally quicker to be developed. And then they work after vaccines are being developed, not as a surrogate for vaccination, but to supplement into populations that can't be vaccinated. For example, a child could get sick with COVID-19 right now and go to the hospital and get a monoclonal antibody, but they can't go get vaccinated. The same for very vulnerable elderly populations and et cetera. And so I think that there is a place in the world for therapies. The difference is that a dose of bamlanivimab is $1,250 and a dose of mRNA 1273 is $30 to the government and free to you. So vaccines are a cost-effective way to prevent disease.
Right.
Therapies rely on people to, number one, have the money, and then number two, to get sick. And if you could just prevent it, why would you even go there?
Exactly. Okay, Barbara.
All right, I'm going to jump in on this one. So this kind of goes with the hesitancy to take the vaccine. Do we have any information yet on the safety of the vaccine in somebody who might have autoimmune disease? Any information, preliminary stuff?
Yeah, you know, it depends on what the disease is, and we didn't enroll people with autoimmune diseases, like I see the question, like lupus and et cetera, into the phase 3 clinical trial. So that's really a kind of an individualized basis. And this is a place where it's good to just say that there are special populations, so to speak, whether it be people who have autoimmune diseases or people who are pregnant, where you have to really do distinct trials for safety in those specific populations. And those things just have not been happening yet. Same way with children, right? And so the short answer is no, we don't know.
Okay, great.
So one of my grade 10 students, chemistry students, Maddie Hannah, is asking an obvious question to her. How soon are we expecting a vaccine for people that are under 18?
Well, the trials have started. Pfizer actually finished enrolling their, I think 12 and up trial yesterday or the day before. So one might expect to see data in those trials in the next few months. And I would expect if they're safe and effective, then the FDA would very rapidly attempt to get those into emergency use as well.
Okay, very good.
All right, so I have another one here. I've seen a couple of these sorts of questions where, individuals are there's questions about why the negative side effects from taking the vaccine. Can you speak to the immune response and what's happening, why people, have the aches and pains and cough and feel like they have the illness after taking vaccines?
Yeah, so I don't call them negative.
All right.
Start there.
There you go.
And I also, actually, to be honest, I don't even like saying the word side effect because that carries a negative connotation. It's reactogenicity. Reactogenicity is your body's natural reaction to a thing. And that's what's happening. The same way that people be like, oh, I got the flu because I got the flu shot. Actually, no, you didn't. What you had was reactogenicity to a thing that entered your body. And it's basically just a waking up of your immune system. Your cells are doing what they need to be doing. There's inflammation, that's where you see the fever, and that's where you see, joint pain and stuff like that. It's inflammation that is an innate immune response that helps to build up what is your adaptive immune response. And so you would be concerned in so many ways if you got a vaccine and you didn't feel anything at all. I do very well with vaccines. I always have. When I get my flu shot every year, I don't even get, I get like a sore arm and then it's like nothing, but I know people who get like chills or whatever else. And so it's just your immune system working for the good.
Okay. So apparently somebody from South Africa, I am a South African American, maybe they're here in America. Anyway, I'm concerned about the variant, my birthplace, South Africa. Will your vaccine protect them?
I'm sorry, I didn't understand the question.
The variant, the variation.
Oh yes, so I talked about that early on. Also, I put the link to that tweet thread into the chat so you guys can check it out. But long story short, we think we're fine.
Okay. Okay.
All right, I've got another one here I'm going to throw in. So the question is, can you explain why the production of the vaccine appears to be so slow? Like, how long does it take to produce? You knew that was coming. I can see.
No, it makes me so sad that people call any of this a failure. Do you know that these companies went on a limb and started producing vaccine without even knowing whether or not it would be bought?
Yeah.
So what is happening right now is life. It's not slow. You can't have expected a company for the last year to produce 360 million doses for the entire company just because it's a pandemic. That's billions of dollars worth of money that could potentially have been wasted. They did what they needed to do. They produced 40 million initial doses, waited for FDA approval. Those doses are being distributed as we speak. And they've promised the hundreds of millions of extra doses by the spring, which is on par with what anyone could expect. It's not slow. It is absolutely on time and very generous, because from a company business perspective, absolutely generous.
And I think actually operationally right now, it's getting the vaccines in the arms. I mean,
it's getting the vaccines in the arms. And yeah, I don't think I wouldn't expect anybody to be producing at risk hundreds of millions of doses of vaccine.
Yeah.
It's unfair to say that.
Good.
So another question that popped up here, Joy Anderson. If you already had COVID-19, do you still need the vaccine?
Yes.
Okay.
The current recommendation is that you still get the vaccine if you've already had COVID-19. And the reason why is all over the literature, in that people, number one, can potentially get reinfected if they've had COVID-19 before, and the antibody responses are not as robust as vaccine responses. That's all over the literature as well. And so these vaccines beat out natural infection, and you want to make sure that you have the tip-top most amount of protection that you can. And you cannot count on COVID-19 or SARS-CoV-2 to give you that naturally.
Okay.
All right. Okay, so this question is with regards to the variants again. Does the development of this vaccine have future import on other coronavirus variants? Like, it seems like, it's a long process. It's years and years already in the making. So it seems like it's an additive effect of what you learn each time.
Right. So you learn more and more each time. I can't talk about what is going to happen for variants. I mean, I can't speak on how evolution is going to happen, but what I can say is that the immune response that's targeted to the spike protein is what we call polyclonal. So a lot of clones of antibodies, as opposed to monoclonal, which is like one clone of an antibody, which would target one space on that spike. So even if you have one antibody that might fall out because there's a variant virus, there's all of its cousins that will bind to other sides of the spike protein and help to really facilitate the type of what we call neutralization or protection that we need. So I can't predict the future, but I think that I wish that there wasn't a ping on people's phone every time there was a variant. I wish that the media would handle variants with a little bit more scientific care, is what I'm saying.
Yeah, yeah. Is there much of a difference, Dr. Corbett? Can you speak to this? Because I know you're representing Moderna right now. Is there much of a difference between the vaccines that are out there?
Yeah, no, I actually don't represent Moderna. I represent the federal government by way of the National Institutes of Health. And Moderna is our close collaborator. But I helped to develop the Moderna vaccine, but the Pfizer vaccine is basically the same thing. The antigen or the spike protein that's being expressed is the same 2 proline mutations that help stabilize it that we published in 2017. The difference and also it uses messenger RNA, and it also uses a fat ball or lipid nanoparticle to deliver that messenger RNA. They're both given into the muscle. They both don't contain any preservatives. The difference is that Moderna uses a 100 microgram dose given twice. Pfizer uses a 30 microgram dose given twice. Moderna's vaccine schedule is 1 dose at day 1 or day 0, and then 28 days later, 2nd dose. Pfizer does 1 dose at day 0, and then 21 days later, a 2nd dose.
Can you speak to the Johnson & Johnson one?
Well, Johnson & Johnson doesn't have one just yet.
No, not yet.
Yeah, and I say that because it's hard to speak about it because they're testing various different they're testing various different vaccine schemas in their phase 3 clinical trials. So they are proclaiming to elicit antibody responses that are robust after 1 dose. So they're giving a spike protein delivered by an adenovirus, which is just a virus that causes a common cold. They're taking out all of the nasty stuff in the adenovirus, like that makes it replicate and all those gross things. And they're putting in the spike protein so that you get the adenovirus and it delivers a spike protein. And so it's a very cool way to alert the immune system of the coronavirus spike protein. They are testing both 1-dose regimens and a 2-dose regimen. And I think that the data just have to be forthcoming around knowing which one will go, or both, that will be approved if approved by the FDA.
All right, Dr. Corbett, we thank you.
Thank you. I'm sorry that I'm generally very oh, I have a makeup.
Oh yeah,
I'm tired. Okay.
I get it. And there are no more questions. There are no more questions that we will ask you.
It's there's a lot of students who are just like, oh my God, I want to go into vaccine development. And I'm like, you know what? It's fun until there's a pandemic. Actually, it actually has been an absolute pleasure. But when you work about 14 or 16 hours a day for a year, now I'm a little bit bored.
Yes.
So I'm sorry if I came off like, oh God.
No, 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 no. It's the kind and human thing to do, you know? I mean, we've been going at this for at least 30 minutes. So
Yeah.
thank you very much for your patience, for your endurance.
Of course.
so
No,
what-
and then also thank you for, having a community facing event.
Yes.
I think that is the duty of academia right now, is
That's true.
to spread our wealth beyond the doors of the halls. So
Mhm. So we'll transition over now to our president, Dr. Loxton, will close us out.
Well, thank you all very much. Thank you so much, Dr. Corbett.
thank you.
This has been really informative. I'm not a scientist, but I understood just about everything, and feel feel much more informed. Feel I can answer a few people better now with the questions that they ask. So thank you very much. There's only one other person I know who's been in vaccine development, and that's someone in Pfizer. And when I talked to them, they said pretty much what you just said right now, that since March, it's just been nonstop. I mean, the hours that people have been putting in and working have just been
I keep saying that. So there's all this talk around igniting a really robust pandemic plan. And I'm saying, I hope that in whatever plan we come up with as a country or otherwise, that embedded in that is that any scientist after the pandemic gets a 1-year paid vacation. I feel like I deserve to be on an island.
It sounds like you really do. And I mean, amazing that you were willing to spend this time with us, because with all that time you're out there doing this and you came to talk to us.
I mean, honestly, it's refreshing to say the least, to speak to people when you are working from home and looking at words on paper all day. So.
Well, good. Well, thank you. And something's going to be coming to you, and I want to show you what it is. We have this little, what we call our presidential medallion.
Yeah.
And we give it to individuals who in the community make a difference. On the back, it's presented to you, and then the words say, "Live today to touch tomorrow." And I think particularly in this pandemic, that is so appropriate when it says "touch tomorrow." And on the front, it says for leadership, compassion, and service. And there's two images here. There's a bridge, which is really bridging over the trouble from one place to another to make that difference. And the other is two hands reaching out. And again, I think very symbolically, they're not quite yet touching. They're wanting to touch, but not quite yet touching, which I think is really what the vaccine is all about. It's enabling people to touch again, to bridge their lives again from one place to another. And we just want to thank you so much for all the energy that you have put into making a difference, all the hours you put in, all the energy you put in, your compassion. Thank you so much for just being you and for sharing yourself and your knowledge with us today. So thank you. And now we got virtual applause. And thank you. And thank you everybody else too for tuning in. You've been tuning in from all over the world, all over the nation, all over Michigan, and we appreciate everyone else coming too. So thank you all, and we're going to close down now. So goodbye everyone, and thank you. Bye.